during PCR era, during genomics era, and post-genomics era. Now the question is how to utilize the genomics data that has been available in public domain and how to use these genomic resources for different purposes. For instance, in plant pathology, you can use this genomic data for developing new genotyping tools. In microbiology also it holds true. And what I am going to do here, how we utilized the genomics data from 2 plant pathogens and how we handled the data sets to address some of the biological questions. The first case study what I am going to discuss with you is about a plant pathogen which is causing a serious disease in rice crop and many other cereals, that is Magnaporthy voriaceae, which is responsible for a blast disease in many monocot cereals. And the title is Utility of Whole Genome Sequence of Plant Pathogens, how this whole genome sequence can be utilized to address some of the biological questions, to know more about this pathogen and how those information can be utilized in crop disease management. Just to give an overview of this particular disease for the information of the trainees, this is one of the globally distributed plant pathogen, and the star, every star indicates that there is a report of a blast disease in those countries or in those continents. You can see here this disease is really widespread wherever the rice is cultivated. And the rice blast disease is distributed globally in all rice dependent countries. It is one of the major production constraint in rice, and it is caused by a fungal pathogen belong to Ascomyces, that is Magnaporthy voriaceae. And you will find more about this particular disease in that particular publication what I put it here, that is Springer Plus, and approximately around 60 to 100% yield loss is reported in many countries. Just to tell you about some fact sheet about Magnaporthy voriaceae, this is one of the fastest evolving plant pathogens. This pathogen is provided with enough opportunities to evolve, and this is reported in many countries that the pathogen is extremely variable. The variability which is present in this pathogen is a simple reflection of the fast evolving nature of this particular fungal pathogen. And it is also believed that this pathogen is evolved in agricultural field, and much like rice, we also started calling this pathogen as one of the domesticated plant pathogens. The reason being, we breed rice to produce more and more yield and more better qualities, and hiddenly this pathogen also evolves to keep pace with the rice development. And this is one of the most challenging disease. Currently, we deploy 2 independent techniques to manage this disease. One is host resistance and another one is agricultural or agrochemicals. And unfortunately, these 2 techniques are not really durable. In particular, the host resistance, which is extremely vulnerable for the so called breakdown, since the pathogen is the fastest evolving pathogen, and the rapidity at which it evolves is a bothering factor in agriculture, because you need to keep this resistance breeding program all the time. There is no stopping for that. And chemicals, of course, again under the scanner, the reason being the produce what is coming out of this chemical based agriculture. In the last few years, the consignments have been rejected by some of the countries because of residues of chemicals, in particular carbon dioxide. the two best technologies what we have, both the technologies are being questioned for various reasons. So, we need improved techniques for managing this particular disease. And some more fact, the conidial germination is very quick, it takes only 2 hours to germinate and often forms an infective aprosorium very rapidly. And this pathogen does not require any natural openings for entry into the host. It can directly penetrate the phyllosphere, causing disease on the foliage. So, that is really a botheration, because this pathogen at any time can make this aprosorium and enter into the so called epidermal layer and can cause disease. And of course, this disease affects both leaf, the photosynthetic area is dramatically reduced, and the serious outbreak of the disease is noticed both in leaf as well as a panicle and neck also. But when it strikes, when the crop is at mature stage, that is the panicle or neck, what is called as panicle blast and neck blast, the yield loss is enormous. So, for that reason, we need to really develop better technologies for managing this particular disease. And what is shown here is a typical neck blast, and what is shown here is what is called as leaf blast. You can see the typical symptom on the foliage, it is a spindle shaped lesion, and what you see in the middle is the sporulating lesion. Remember, the lesion which is sporulating is very dangerous, because this conidia can easily move from one leaf to another leaf, one plant to another plant, to another field, and even one region to another region. We kept some biological questions in our mind, and we wanted the genomics data to address that. And these are all the questions, and this is also very important for you. Before taking up any genomics related project, or you wanted to use the genomic tools, ensure that you have a biological question in place, and that biological question needs the genomics intervention. This is very important, right? The biological question what we kept in our mind is how far the strains which is prevalent in our country is genetically close to the strains reported elsewhere. And the question number 2 what we had is very pertinent question. Many times scientists debated about it, there is no conclusive answers, that is, what is the relationship between leaf blast and neck blast. The reason being, these two blast occurrence happens in rice in 2 different time points of rice life cycle. And whenever you see leaf blast, which happens when the plant is still in the immature stage, the yield, the crop failure is noted, and people have reported when the season changes and the rest of the plants escapes and it still survives, and the panicle comes, they do not see any panicle blast at all. So, scenario number 1. So, there are serious incidents of leaf blast. In those fields, the panicle blast was reported very intermittently. Sometimes there is no panicle blast. And even more intriguing question is, there is no leaf blast at all, but all of a sudden the panicle blast comes and kills the plant. So, the relationship is always questioned, whether it is by a same strain or it is by a 2 different strain of blast which is causing the panicle blast and the leaf blast. So, our second question was, are they genetically similar or are they different? If they are different, how far they are different? And third one is, can we develop a specific marker? Of course, that is the reflection of the question number 1. So, we wanted to address that one also. So, we started this project, what is called as Magnapurthy Genome Project, thanks to ICR for funding this project. And the strain what we have taken on board is one of the Magnapurthy strains which was isolated from Bihar and which is able to knock down 24 well recognized R genes of rice. So, we called this strain as one of the most virulent strain, just because it is able to knock down 24 R genes. And not only that, this pathogen is able to cause lesions on some of the cereals which is not really close to rice. For example, upon artificial inoculations, we could find that this rice plus strain is able to infect one of the most important cereal in northern plains, that is wheat, barley, oat. So, this is another reason why we selected this strain for whole genome sequencing project. Just move on. This is the fact sheet about the strain what we have elevated to genome sequencing project. Remember, before you take up a genome sequencing project, the relevance of your organism or biological material is very, very important. The biological relevance is very important. And I already told 24 R genes are knocked down by this strain, except two strains, two R genes. One is PI54 and another one is TITUP. We have gone into a hybrid approach. What I mean, this morning we were discussing with the nanopore technology, the technologist who gave a lecture here, and he also emphasized we need to go for two different technologies to complete the genome sequencing projects. So, we were comfortable in handling two independent sequencing technologies. One is the short read technology for the benefit of students. Short read technology provided by a company what is called as Illumina, that is HiSeq 2500 was the technology what we used. And the long read technology, it was rolling at that point of time, that was 2015, 2016 time, that was PacBio RS2. So, just to give you a little more feel of the genome sequencing technology per se, the machines are able to provide you the reads, sequencing reads. The length of the read depends upon the chemistry and the technique what you are using. HiSeq reads are very short and PacBio reads are very long. So, it is something like this. The reads are provided to you by the machine, and that is assembled into slightly longer one, that is called as contigs. Contigs is a kind of it is not acronym, it is a shortened form of contiguous sequence. Okay. Two short reads are extended by having the overlap, it becomes longer one, right, and this longer one is again overlapped with another two, it becomes still longer one, right. So, the two contigs are again made into even bigger, that is called super contigs, right. And yesterday the speaker was beautifully explaining the super contigs can be made into scaffolds by putting some ends. So, that is scaffolds, and finally, the draft genome is made. The manual what we prepared describes all these terminologies. Please read that and you will get more about it. Reads, contigs, super contigs, scaffolds and draft genome. And I did not put complete genome here, that is altogether a different technology technology. You need to have opgen mapping, you need to close the genome, that is ultimate. And most of the genome what is published so far is draft genome. I am not getting into the chemistry part of it, which is explained well by different speakers here. The PacBio yesterday it was spoken here, nanopore spoken already today this morning, and somebody will be speaking about Illumina in the coming days. This is what we proceeded further. After isolating the genomic DNA, we prepared true sec nano DNA HT library that is prescribed for HiSeq 2500 machines for reading the DNA or genome, and this is 2 into 125 base pair chemistry, and we could generate around 10 GB of data from this chemistry. And PacBio Bell library, yesterday it was discussed here, a hairpin adapted protocol for long ultra read sequencing on PacBio RS2 platform. We could generate around 500 MB data in one smart cell, and we used P6C4 chemistry in long read sequencing by PacBio. This is a standard pipeline many companies gives you. I just took it from the report, and this is how the whole genome sequencing project is carried out, right. It tells you different methods, the quality checks, the assembly, and of course, after assembling the genome, we will be predicting the coding regions, also called as CDS or gene prediction, whatever it is. Then we go for annotation and number of other analyses what we wanted to do to address the biological question. This is a standard pipeline. Most of the genomic service provider will tell you, but I must tell you here, you must discuss each and every point with the private person or wherever you want to do the sequencing of your samples. It may be a private or it may be a public funded institute. Wherever you go, try to discuss every point and what it really mean to you, that you to know. And we have handled two different technologies, a short read 2 into 125 base pair, another one is a long read from PacBio. So, two sets of data is in your hand. Now, the question is how to use both the data sets, so that end of the day you have one data set which we would like to call it as draft genome. So, at that point of time, the algorithms were still evolving and there was no technology or no pipeline to use these two data sets. One is a short read, another one is long read. So, we used a specific assembly program what is called as SPADES. SPADES is an acronym, St. Peter's Assembly. You just Google it out, you will get more information about this, right. So, the SPADES program, it accepts long reads and short reads, and you can use both to stitch your genome up to draft genome in a simple language, right. And it is typically a bioinformatician's job. So, Rakshit was talking about random error, and this morning we heard about systematic error in short read technology. And these two errors can be addressed when you go for hybrid assembly. The random errors will be corrected by the so called short read, and systematic errors is corrected by the long reads. So, this is the perfect combination of two complementing technologies. Each technology has got some deficiency. One is having systematic error, another one is having random error. When you merge these two, and both errors are removed, and we come out with a kind of genome assembly. And the genes are predicted by number of programs. One such program we used here is Augustus program. Please read more in the either in the manual or it is in the Google, you do it. And of course, we uploaded the data in GenBank. And it is already pointed out the other day, once you upload in any one of these databases, within 24 to 48 hours, the data will reflect in other data centers also, and that is what exactly happened here. Yeah, to tell more about the genome of the pathogen, which is able to knock out 24 R genes. This is a statistics. This genome is already published in GenBank. This is what is called as the accession number, which is provided. It is in public domain. Anybody can access this data. And the genome, we also faced a lot of issues and problems. We could solve it. Just for the information of the house, when it was originally assembled, we could get the assembly done in 2500 scaffolds, or I would say 2500 contigs, right. So, the genome size is around 35 MB. The number of contigs is 2500 contigs. That means, a 35 MB of genome is 2000 equal to 2500 contigs. In other terms, 35 MB genome is represented in 2500 contigs. Here comes the problem. When we approached a publication by Genome Announcement, that is one of the top end journal from American Society for Microbiologists, ASM, they referred back to us, they saying that your assembly is not acceptable. The reason being, the genome size is only 35 MB. A 35 MB genome in all standard is not a big genome. It is coming under the category of small genome. Dr. Kishore might have talked to you about the small genomes. So, they wanted us to reassemble the genome in less than 1000 contigs, or as minimum as possible, right. The best assembly in the world is single contig, that is rarely achieved, right. It is rarely achieved. A single contig, the entire genome, right. So, we reassembled the genome once again to meet the international standards required for research paper publication. When we did the reassembly, we got the entire genome represented in 996 contigs. The number of contigs have come down 2.5 times, and they accepted the paper, right. So, it is always better to approach a specific publication platform to assess the quality of work what you did. Many times we feel, okay, it is all done, that is what we felt, but end of the day it is not so, you need to redo. The reassembly, it is nothing but running the algorithms over and again, eliminating the unwanted repeats, unwanted sequences. Assembly is really challenging in any genome sequencing project. That is what everybody is talking about, this assembly. Now, the OpGen mapping has come to close the genome. Assembly is not that easy, right. Yeah, this paper is published in Genome Announcement under the title Genome Sequence of Unique Magnaporthy RMGDL isolate from India that causes blast disease in diverse cereal crops obtained using PacBio single molecule and Illumina HiSeq 2500 sequencing. The whole genome is assembled with a minimum number of contig is one of the requirements for any whole genome sequencing project, whether it is a smallest genome of your virus or it is a bacteria or fungi. You should assemble the genome in minimum number of contigs. That is closer to the reality, right. If it is a single contig, that is the best, that is never achieved in any organisms. Some people started claiming 2 contigs, people are assembling bacterial genome nowadays. So, the exceptional cases are coming now, right. And microbial resource announcement present, it was now the renamed the journal. Originally it is Genome Announcement. It suggests that fungal genome should be assembled in less than 1000 contigs. So, then only they are going to approve or accept your manuscript. This is some statistics for the benefit of all of you. Maybe you will find some jargons also here. So, the number of scaffolds, or I would call it as a contig here, 996. This is a genome size, roughly around 35 MB genome. The average size of our contig is around 16,000 base pair, and N50 scaffold is around 45,000. N50 value is an indicator that 50% of the reads are in excess of around 45,000 base pair, and of course, the minimum contig is around the maximum size of the contig what we got in our pool of reads, that is around 186 500 base pair, and the minimum one is 500 base pair. The entire genome was now run into Augustus program to know how many genes it is capable of coding. We could see around 12,000 plus genes, and the average gene length is around 1000 base pair, and we have biggest gene maybe 14,916 base pair, and the minimum one is around 201 base pair. These are all predicted from the Augustus program based gene prediction. Remember, this is only a prediction. If you want to really know these genes are existing in Magnaporthi, what is that you are supposed to do? Anybody? Before that? Anybody? This is predicted number of genes of Magnaporthi, which is around 35 MB in size. If you want to know, yes, this genome is capable of producing this many it is capable of producing this many proteins. What is that you are supposed to do? Transcriptomics. The data I did not bring it here. We have done the transcriptomics of this Magnaporthi when it is in the developmental phase. For example, Conidia to mycelia, mycelia to again Conidia. You got it? That you put the two phases. Phase number 1, when the fungus is germinating right from the Conidia to the mycelial biomass, right. So, we have developed a kind of a simple mycological intervention in the lab, collecting the RNAs when the fungus is still in the mycelial phase, but there is no sporulation phase started. Second phase is it is profusely sporulating. So, the two developmental phases you collect the RNAs and go for transcriptomes, that is also done, and the data is closer to what we got here. Of course, slightly less than this rate, but is almost closer to this figure. Any questions? Yeah, of course, yeah, that is true. We might have definitely missed. You should infect the host, then again you have to go for transcriptomics of those host material, and removing the host transcripts, take out only the pathogen transcripts. That can be done. That is what I already told. We less than what is predicted out of this Augustus. Maybe the missing ones are the one which is expressed when the pathogen is in the infective stage on the plant. Yeah, this is just to show you how this SPARDAS front page looks like. This is St. Peter's Academic University of Russia. That is what the St. Peter's, right? And this is how it looks. If any one of you wants to know more about this particular program, please get into this website. That takes you take a tour of it, and you will understand what this particular Spades program is capable of doing. This is a little bit more about the statistics only. I will just move into. This is gene prediction we did. This is a front page of Augustus gene prediction program, and you can see that you have to run this program to predict the genes from your genome. That is around 12,747 genes we predicted. This is some of the pathway analysis. This is beyond the scope of this class. We will just shift it. Yeah, this is what Kavi was asking, the genes which is expressed when the pathogen is in the infective stage. If you want to know, there is a specific websites and specific portals are available nowadays, what is called as PHI base, that is pathogen host interaction, database. So, this is another wonderful database to know which of your pathogen protein is participating in host infection. In nutshell, it will tell you how many of the proteins which is expressed in this pathogen is having the function of effectors or having the function of a protein which is participating in virulence. So, those information you will get from this particular website. That is what the virulence function, effector function, and different parameters are there. You will be able to use it. So, once you maybe you will when you start using this kind of work, you will understand the intricacies of this database. right. One of the hallmark of this data set which we generated from the whole genome sequencing is large number of what is called as TE, that is transposable elements. This came as a kind of offshoot data from the whole genome data. So, you can see here different categories of transposable elements. Several hundred transposable elements were found in the genome, and of course, these transposable elements are so important for fungus to evolve time and again, over and again. I already told this is one of the fastest evolving pathogen in agricultural field. Maybe these transposable elements directly contributes towards evolution of different virulence genes, evolution of different pathogenicity genes, or maybe it helps the pathogen to adapt in different niches and different environment conditions. So, I do not want to read it here, starting from cacta, crypto, so many transposable elements could be found in the genome. Yeah, this is a front page of our genome data, that is in GenBank. Now, let us come to the first question. How far this genome is genetically close to the genome of Magnaporthy, which is causing disease in another country, maybe causing disease in another host, right. So, only these two things. Maybe Magnaporthy is already told it is reported in most of the rice growing continents. This disease is reported right from South America, Africa, India, right here Southeast Asia, China, Japan. The problem is everywhere. And the question is whether our strain, how far it is genetically closed. So, what we did, the assembled genome was compared. The assembled genome, which is around 35 MB in size, is compared with 15 other genome, because at that point of time we had around 15 genome data was in public domain. If you look at today, in 2 years of time, we have around 212 genome of Magnaporthy. In 2 years time, the database has increased dramatically from 15 genome to 212 genome as of now. I will just quickly go through it. The actual molecular phylogeny part I am not describing here, that was already handled, right, by Dr. Anirban. So, what we did here, the 35 MB of genome is subjected to multiple alignment with 15 other genomes, right, and the 15 other genomes we got it from different countries. For example, we got it from mostly from Brazil, because in Brazil there is a serious disease in wheat that is called as wheat blast, and so many genome was sequenced, right. So, those genomes we have taken on board, plus some rice genome, rice Magnaporthy genome also from India we have taken. And this is I just draw your attention, this web page has got most of the genomes. I can share this web page, link with you, and this is the kind of data what we could generate, and our genome is somewhere here, and 1, 2, 3, 4, this 4 is from rice. Okay. For the information of pathology students, 7015 strain is a first Magnaporthy strain ever sequenced. The paper appeared in Nature, author is Dean et al. That was a laboratory strain, unfortunately. There was no, when we trace the origin of this particular isolate, it is rated as it's a laboratory strain. That means somebody was using as a kind of model system in their laboratory, they sequenced it. Rest all 3, these 2 are from India, right, and this is our genome. And come to the next one, we could see the 15 strains what we handle, the whole genome multiple alignment. After doing the multiple alignment, you come out with a visualization of the data. This is a visualization of the data, and you could see roughly 4 major clusters, right. And these 4 major clusters the first cluster, all of them are the strains which is causing wheat blast, right. And we have somewhere in the middle, 2 of them are wheat blast and 2 of them are ryegrass blast, and later on we understood that these 2 wheat blast and ryegrass, they are cross infecting each other. That information later on we got it. And this is from Indian goose grass blast, it is from grass isolate, and this is rice isolate. So, the data what we got from multiple alignment of the whole genome is nearly matching with the phenotypic observations one can compare. For example, all of them are from wheat. Here indirectly speaking again from wheat or ryegrass, but they have the cross infectivity with each other. This is of course, it is a single isolate. We do not know if one more grass is taken, how the tree would have disturbed. We was talking about the other day, and this is rice blast. So, the biological question what we had kept in our mind, how genetically it is close to rest of the strains. What we understood is it is distant from the other strains which is causing blast in other crops. So, that is a very important answer we could get from this particular whole genome sequence data handling. This is South American wheat isolates, this is rye isolates and wheat isolates, they are cross infecting each other. This is grass isolate, this is typically rice isolate, and remember the 7015 strain is not an Indian strain, it is an American strain, a laboratory strain, and yet it is able to form a single cluster with rice. So, that means the rice infecting isolates are genetically close to each other, right, and the wheat infecting strains are closely related to each other. So, that inference we could make. Now, question here is if you include more entries here, how this tree will change, that is beyond our prediction, right. So, this is I skipped this slide, it is not needed. Actually, we compared our data with the outbreak of wheat blast that happened in Bangladesh, and we could find that it is completely different from the strains which was causing wheat blast in Bangladesh. So, that data we could generate here. The findings what we could make is the Indian strain of Magnopurthium rice infecting rice and other cereals including wheat under controlled condition is genetically distinct from the one which caused blast epidemics in South America and Bangladesh. The second biological question, I will just move on to the second biological question, that is can we observe the genetic variability in Magnopurthia at intra-field, intra-seasonal scale? For example, if you make a collection of these isolates in a single field, in a single season, can we find a kind of genetic variability? It is a very interesting question we wanted to address. And if you happen to isolate a rice leaf blast and panicle blast within the season and within the field, can we predict or can we understand the genetic similarity, right? What is more important here is the collection from the single field. The single field which includes a neck blast and also leaf blast, right. So, we had a huge collection of 60 isolates, only we handled a few of them in the analysis here, and the genome around 26 of them we handled in the laboratory. The genomic DNA was isolated, and isolates which represent leaf and neck blast, that is from a Kashmir valley in a single field we collected it. Monosporidial isolates were established, and this is the utility part of the whole genome data, right. What we did, since we had the whole genome, so we handpicked a number of genes from the genome to make the assessment of the genetic variability, right. So, through an approach what is called as multi-locus sequence typing. So, this is what I am going to teach you. This is a lecture, maybe after the lecture I can tell you how exactly this is conducted in a laboratory. Okay. Of course, we have selected 18 genes from the genome, and 11 of them are the housekeeping genes and 7 of them are virulence genes what we handpicked from the literature. In total 18 genes were shortlisted for this analysis. And we have included 2 more isolates, which is also very prevalent in our country. One is finger millet, another one is pearl millet. The blast in finger millet and blast in pearl millet is also a serious problem in those respective crops. And of course, 11 housekeeping genes and 7 effector genes. This is a pipeline. So, you have the genomic region was selected. We have decided the 11 housekeeping genes based on their conservation level, and of course, we have selected 7 virulence genes from the literature. We have designed primer using the same protocol what was handled in the class yesterday, using primer 3+, the parameters, everything was conducted as per the class what we did yesterday. We did the PCR amplification, and for you, if you want to go for cloning option, you can do that, but we did the PCR amplification, sequencing of target genomic regions of those 18. We did the sequence analysis, sequence curation. Today we will have a detailed discussion about that, and the conserved consensus sequences was retrieved, and that was converted into a molecular phylogenetic tree, and we could assign some allele numbers also. What is more important for all of you is how to convert the sequence information which is coming from the consensus sequences into a molecular phylogenetic tree, number 1. Can we do this analysis in a different way, right? That is through assigning the allele numbers after observing or after finding out the single nucleotide polymorphisms in the sequences. In few slides, I will show you how it is done. So, this is a sequence based phylogeny, another one is allele number based population genetics snapshot. So, this is you need a little time to understand how these concepts work here. We will take small example how this is the view of the amplicons of all 11 housekeeping genes and 7 effector genes, or the genes which is participating in virulence. This is a pipeline. This is the GenBank number of the combinations. We handle around 24 isolates altogether. This is 11 housekeeping genes, and each one of the gene is sequenced, curated, and best reads were retrieved. We prepared the so called data sheet, submitted in GenBank, and GenBank number is assigned, right. So, it is a massive work in terms of sequencing, curation, submission, getting the accession number. This is a snapshot of all those 264 housekeeping genes representing 24 isolates were generated and published. And also the 7 effector genes we got from literature. The same story was done here as well. Around 168 effector genes representing 24 isolates were generated and published. So, now we have the data sets. Remember, these 24 isolates were now characterized just by using 18 genomic regions. I will put it in that way. You understand? So, why we did that? We could have gone for sequencing all those 24 genome also, and considering the cost involved, the time involved. So, we opted for this tool. So, this is one of the fallout of the whole genome sequence data. You can develop a genotyping tools from the whole genome data to address some of the biological questions. What I mean to say is, it is always not necessary to go for whole genome data. You can go for a technique what is called as resequencing of a specific genomic region. That is what. You can do the resequencing of whole genome also, provided your time and the cost allows. So, at that point of time, it was too expensive for us to do the sequencing, because two different approach, sequencing cost a huge amount of money is involved. So, here you do not need that much, a fraction of that money is spent here to generate the same information which is going to address some of the biological questions. So, this having done that, this is a 24 isolates representing a single field, and it also represents a leaf blast and a neck blast. And you can see here what is L, that stands for leaf blast, and what is N, that stands for neck blast. The picture very clearly sees that we could see extremely minor variation in the population in 24 isolates. right. What happened here? All of them are clones of each other, almost they are clones of each other. In particular, the leaf blast and neck blast is sharing all 18 genes or the sequences of all those 18 genes. You understand? There is not even a single base pair variation. So, that prompted us to make a speculation that these two are one and the same. There is no genetic variability between leaf blast and neck blast at all. A subtle variation we find in another clade, and you find it in leaf blast also, and we could never see neck blast is coming separate and leaf blast is coming separately, never. So, some islands of grouping comes here, but again the quantum of genetic variability is extremely minor, right. So, that prompts of course, you can see here that is well supported by the outgroup. PL millet is completely different from rice blast, right, and the finger millet is maybe a little closer to it, right. So, you can make lot of other interpretations, right. The genetic relationship between rice blast strain and finger millet strain is very close as compared to the genetic relationship between rice blast and PL millet blast, right. The PL millet blast is a completely different genetic lineage. So, this information was skipping genes, and this is a tree what we could construct, right. Yeah, this is a clade. The key points: MLSE based genotyping clustered all rice isolates in one group, with the finger millet and PL millet isolates clustered separately. No genetic difference between neck blast and leaf blast could be observed. Intrafield genetic variability is also found, which is very, very extremely minor. That also make a strong prediction that this strain is a clonal, which is making rounds in agricultural fields and causing disease. Yeah, I will stop it here. I will come back to you once again for the second part of the lecture, that is how to convert the sequence polymorphism into allele numbers, right. Maybe I need 1 hour time to teach that particular story. That is very, very important. That particular approach can accommodate several hundreds and thousands of entries in a single population genetic analysis. Remember, the classical molecular phylogeny has got a limitation. This is 24 isolates. It is already covering the entire slide. Suppose if you are handling thousands of entries and it is going to be too crowded, the visualization tool itself is the one which you cannot appreciate. Now, we have modern tools which can accommodate. We have constructed a phylogenetic tree with 1,000 entries of isolates, right. Still the information is coming out tangibly. You can understand what the tree speaks. That is possible through a completely different algorithm, what is called as E-bust algorithm. This E-bust algorithm, the input data is allele numbers. These allele numbers are assigned for specific nucleotide polymorphism, which you observe in your multiple alignment analysis. So, that portion we will take it up in the coming days. I have a series of slides on that, where we are assigning the allele numbers based on single nucleotide polymorphism, right. So, I will stop it here. Any questions from anybody? Yes, please.
What is the reason behind choosing virulence genes?
It's a very interesting question. Most often the housekeeping genes alone will not reveal the pathogenic behavior of your pathogens. You need to necessarily accommodate the virulence related genes also, although it is also evolving very fast, and you know it very well. The virulence genes evolution is a single point mutation in those effector genes. Those single point mutation also we need to track to keep an eye on how the virulence is spreading, right. Remember, whenever you do multi-locus sequence typing, never ever practice with only housekeeping genes. You need to take sufficient number of virulence genes to develop actual story what is happening in the fields, right. So, that is very important for us to track the pathogen behavior. If it is a simple microorganisms, perhaps you can even do it with a housekeeping genes, and there also if you keep a particular functional trait in your mind, of course, you have to keep those sequences which is coding for those particular function. So, it is always the combination of housekeeping genes and the functionally important genes to draw conclusions like this. Anybody else?
In initial slides, there was mentioned 2 into 1 to 5 base pair chemistry.
Meaning is so 125 base pair is red. The reads I told now one of the slides, the reads are only 200 to 250 base pair reads. That's it. Short reads. That's what short read technology. Short read technology. Yeah. There only the speakers talked about systematic error. You know. Yeah.
And second, sir, since in this study it is coming up like wheat, finger millet, and this pearl millet isolates are very much distinct.
Distinct. Yeah.
Can you say that cross, infection chances of cross infection?
In fact, deliberately I did not bring all those data here. My colleague Dr. Prakash is working here, and he has done a very extensive inoculation studies. He put the rice isolate on finger millet isolate on rice, again fuel millet on finger millet on rice, all possible combinations. He is telling very interesting observations. These three strains are not able to cause disease on other host. That means rice isolate is always causing disease on rice only, but it is not causing any disease on finger millet and fuel millet, and the story is same with other 2 isolates also. But interestingly, all the 3 strains are able to infect other cereals. That is even more complicated. So, when he inoculated all these strains individually in different hosts, for example, wheat, barley, oat, and so many other things, he is getting infection, right. So, that supports our molecular phylogenetic tree also, because we got 3 distinct clade and each one of them represent the host origin, right. That is more important. So, what we started understanding is the clustering what is happening based on the whole genome data is indirectly indicating from which host the pathogen is isolated from. So, that data is coming, okay, because I showed in the first molecular phylogenetic tree based on whole genome, all rice clustered together, right, all wheat clustered together, in between some mix up, but the other data came, it is infecting. So, the point is the pathogen which is fast evolving, it is always very difficult to predict the genetic variability, because it is all the time evolving, right. So, we can always say that at this particular point of time this is a variability status, right. After some time, if you include, I will tell you in future, maybe tomorrow's class we will talk about it. The allele data speaks almost same story only, but in a different much in depth data is coming out using this MLST EBEST based molecular phylogeny. Anybody else?
If an organism has 7 chromosomes, we can expect the context to be like minimum context to be 7. In this case, how we can expect?
Yeah, that is a wishful thinking of all genome sequencing project, but rarely it is achieved. Magnapurthi it is achieved, it is 70-15 strain. If you are anybody is updating on a regular basis, originally it was only a draft genome, later on it become complete genome. Now it is chromosome wise they mapped everything. So, that means Magnapurthi has got 7 chromosomes, and each chromosome we know the sequence. Maybe somebody might have done karyotyping and they have compared both the data sets, they mapped all those reads in different chromosomes. Now the data is available chromosome wise. Chromosome 1, what is the genomic content, how many genes are there, and so the chromosome 2, 3, 4 up to 7. That is a wishful thinking. Maybe technologies are coming now, the long read, what is that, nano bio something he was talking this morning, that opgen mapping. So, all these things will converge and give you a better assembly and better data, right. Everybody is trying to reveal what is there in nature, correct. There only the flaw comes, even here also genome sequencing also. I told him the best assembly is a single contact. So, that is rarely achieved. Anybody?
You have done the e-burst analysis for the single field isolates.
Yeah,
or
we have done it.
clustering the
Yeah.
tree.
Yeah, we have done it. That maybe tomorrow we'll
No,
take up the
just
discussion.
one.
Yeah, we have done it. They are all, they are very genetically few base pair variations. Yeah. Maybe to get the, this is how it is done. I don't want to elaborate that because time is running out. This is how the allele is assigned here. The data, the visualization will be something like this. This is the allele numbers. The beauty is you can accommodate as many isolates as possible. We have constructed a molecular phylogenetic tree which shows around 212 isolates. This is a molecular phylogenetic tree. This is not possible by a conventional phylogenetic tree which uses sequence data. But here it is not a sequence data, it is allele data. The logic is the two of the isolates, they share all the alleles, they are identical twins. You understand? If all the alleles are shared by, for example, we have 18 alleles here, right? If two of the entries are sharing all the alleles, that means what? There is no sequence polymorphism between those two, right? So, that means they are duplicates in environment. You understand? If any one is evolving or it is deviant based on a single nucleotide polymorphism, that takes different number. For example, here for calmodulin it is 1, for another isolates 8, 8, 3, it is 3. The question is you should know how the number 3 has come here and what is the science behind assigning a number 3 here. That only I want you to learn, right? That I will take up in the maybe next week. We will take up that particular class. It is a simple concept you should understand. Assigning these numbers for those individual sequences, how this number 1 is assigned and what are all the criteria, and how another isolate we got number 9 we got, and how this number 9 is assigned. So, we will take up in future classes. I think otherwise we will run out of time this morning.
So, even though there is no genetic difference between leaf blast and neck blast, and what makes the difference to get only in some condition we will get only leaf blast and some condition we will get only neck blast?
That is something to do with the environment. We all know that environment also 30% role it plays in any disease outcome.
Even sometimes even environment also same conditions, that time also we are getting difference.
Perhaps see, one of the mysteries here is we have not studied that, honestly speaking, right? It is everybody is questioning this one. This has happened in many places. In a particular year only leaf blast is there, there is no neck blast at all. In another time point only neck blast is there, there is no leaf blast or very minimal incident of leaf blast. We have not studied that. I do not think any such data is available. What was the reason for this phenomenon? I would attribute it to only the environment, because pathogen is everywhere. Maybe, yeah, host nutrition might play a role. It's a mystery, because a few years back there was a paper. The paper is all about the neck blast is caused by a completely different developmental stage of this magnoporthy, not the macroconidia. For the information of the other students from microbiology, this magnoporthy produces two different kinds of conidia. The macroconidia, the 3-celled one, is responsible for leaf blast. Now, the Nature Communication paper says there is another form of conidia, what is called as microconidia. This microconidia is something like an endophyte in rice, and it travels all inside. When the rice matures, maybe some virulence genes are expressed and it causes neck blast. But subsequent to that paper, we have not seen any paper which is supported the previous paper. Maybe nobody is addressing it. So, this is still a mystery, but the fact remains, whether it is a microconidia or it is a macroconidia, based on our results, we are speculating that it is genetically identical. Okay. The infection process may be different, and the reasons for the endophytic colonization may be different. So, it may not happen all the time, maybe in those seasons it might have happened. So, the microconidia is microaerophilic and infection is through root. That is even more strange, because we all know that magnoporthy is adapted on phylosphere. But that paper says, not only that paper, another paper appeared in Nature in 2004 by Anne Sesma from Spain. That paper says Magnaporthy has got the ability to infect roots. So, it displays the root infecting fungal behavior. So, that means many things we did not really understand about this pathogen. So, that is the reason why we are not able to answer this question, why this erratic behavior of this pathogen in the same field, right. Yeah, we will stop this discussion. At least one student will have a presentation today, possible. Yeah, I will stop it here. We will continue to discuss this matter, but the point here is, forget about the story what we highlighted here. The techniques which I am highlighting here, that you are supposed to learn, that is more important, right. Maybe in the coming days we will take 1 hour time, we will discuss how this allele number based molecular phylogeny can be conducted by all. It is a very simple tool, we will discuss that, okay. And I will stop it here. Thank you so much for your attention.